All right, so talking about intra-microbiome associations, networks, correlation, and compositionality. So why we build interaction networks to detect co-occurrence, co-exclusion, and modules, which helps us identify community structure, potential functional groups, keystone taxa, and dysbiosis signatures. So moving on, we basically have a simple pipeline. First, we build the detection or relative abundance matrix to check the presence or absence, then infer the pairwise or higher order associations with the statistical methods, and also construct the graph where nodes are taxa, edges are basically the significant associations, that is positive for co-occurrence and negative for exclusion. Now we have pairwise methods and their caveats. So simple correlations like Pearson for linear and parametric, Spearman for rank and non-parametric, and Kendall for pairwise concordance, which is robust to ties and sparse data. So all yield correlation coefficients are belonging to -1 to 1, and p-values against null hypothesis are equal to 0. Spearman is often preferred over Pearson for non-normal microbiome data, and Kendall handles ties or sparsity better. Now we have the compositionality problem, that is relative abundance data are compositional, that is we have a sum constraint. So spurious correlations arise. An increase in one taxon necessarily reduces relative abundance of others, even if the absolute abundances are independent. This leads to false positives. So we have corrections and specialized methods like reboot or CREP, that is renormalization, permutation, bootstrap. Renormalization, permutation, bootstrap, which creates compositional null distributions by permuting and renormalizing data. Creates compositional null distributions by permuting and renormalizing data, then uses bootstrap tests to find significant edges that exceed the compositional expectations. It is useful for pairwise correlation correction. Then we have sparse CC, spike EC, and conet LSA others. There are some other methods which we don't need to get into the depth of. Now moving on, centrality measures. So degree centrality is basically the node degree, that is number of edges. When we have high degree, we have high hub taxa. And we have betweenness centrality, that is the number of shortest paths passing through a node. It identifies the node's bridging modules, which are potential keystone connectors. How do we detect modules? We have the walk trap algorithm, that is random walk based clustering. Nodes sharing any random walk paths are grouped and good for modular structure and structure detection. Edge betweenness, remove the high betweenness edges iteratively to reveal the community splits, yields hierarchical community tree. What is the biological use? So hubs and modules can indicate functional guilds or therapeutic targets. For example, in hubs in disease associated modules, always validate functionally pathways, metabolite associations before intervention claims. Now moving on to multivariate association, where we'll be discussing Permanova. So it basically tests whether centroids or groups differ in a distance matrix. So the full form is permutational multivariate analysis of variance. Right. We first basically compute the pseudo F statistic, that is between group sum of square distances upon within group sum of square distances, which is scaled. Then we calculate the p-value by permuting sample labels, for example, for 1000 permutations. And Permanova can be confounded by unequal dispersion. So we always test homogeneity of dispersion before interpreting group differences. Next up, we have co-inertia analysis, that is multivariate method to find covariation structure between two data sets measured on the same samples. For example, species composition and environment variables. Co-inertia is basically the sum of squared covariances between pairs of variables after ordination. It basically tests significance with permutation using the RB coefficient, useful to find joint patterns of variation. Then we have Procrustes analysis, that is for shape alignment, rotate and scale one ordination to match another. Measures congruence, so it is based on shape correspondence. Then we have WGCNA, which is weighted gene co-expression network analysis, that builds assigned or unsigned weighted networks from correlation matrices, chooses soft threshold power beta to approximate a scale-free topology, computes the topological overlap TOM for module detection. Then we reduce the features to model in eigengenes, PC1 per module, and find module trait associations. Then identify the hub drivers with high module membership and gene significance. And importantly, WGCNA can integrate microbiome taxa and host omics into the same analysis. Right. Now we have confounder analysis. So to test whether an association between microbiome and trait holds after adjusting for confounders, right? Moving on, we can just skip this part. So explain, I asked GPT to explain strain plan in total depth. So strain plan is basically a tool inside the metaflan or human and ecosystem, which is used to identify and compare strains of microbial species directly from metagenomic sequencing data. So we basically can think of it in this way. Metaflan tells us which species are in the sample, and strain basically tells us which strain of that species is present and how the strains differ across samples, the time points, conditions, or people. It's one of the most important tools in microbiome research because species level information is often too coarse. Strains differ in virulence, metabolism, resistance, pathogenicity, etc. So basically, species detection becomes easy using strain. Now, the core idea is Metaflan has a large database of clade-specific marker genes for every species. These marker genes are unique to that species, but present in all strains of that species. However, small nucleotide differences, SNPs, inside these markers can distinguish the strains. So StrainPlan basically extracts these marker gene sequences from our metagenomic reads, aligns these marker sequences across the samples, and builds a phylogenetic tree to show how the strains are related. Now, Metaflan identifies which species are present. So we first run Metaflan on each metagenomic sample, that is the step one. So Metaflan outputs the species detected, their relative abundance, and importantly, which marker genes are present. These marker genes are likely sequence beacons unique to each species. If a species is not abundant enough, its markers may not be covered, so you won't be able to analyze that strain. So we basically have this step one where Metaflan identifies which species are present. Then in step two, we extract the marker sequences from each sample. So from SAM to FASTA. So for each species, StrainPlan gathers all the reads that map to the marker genes and reconstructs the consensus marker sequences. Right. Then step three, we filter markers based on coverage and quality. Step four, we perform multiple sequence alignment (MSA). Step five, we clean and trim alignments, and then we finally, in step six, build the strain phylogenetic tree. Step seven is basically getting the biological interpretation from the phylogenetic tree. So strain sharing, if the samples are clustering tightly, that is the same strain. It is useful for FMT engraftment tracking, household or family transmission, hospital outbreak tracking, and longitudinal microbiome stability. Right, and basically we can check for the important parameters. We have marker in n samples, that is minimum number of samples containing the marker, ensures comparisons are meaningful. Then minimum read depth, minimum depth to call base, trim ends, remove noisy ends, and allowed mismatches, another parameter which filter markers with too much variation. Right. Now we can see the strengths and weaknesses. So in this strain plan method, we basically have some advantages like no assembly is required, accurate for dominant strains, fast and robust, great for population level or FMT studies, and uses trusted marker genes databases from Metaflan. Limitations are needs decent coverage, cannot detect low frequency strains, cannot analyze species or missing markers, and works mainly on bacterial species. Right, now moving on, Panflan comparison with it. So both tools belong to Metaflan or human and ecosystem, and both aim to analyze strain level variation from shotgun metagenomic data. But they're fundamentally different in philosophy and method. StrainPlan is basically sequence-based marker SNP variation, and PanFlan is pan-genome gene presence or absence variation. So the questions that they ask, firstly, StrainPlan would ask, are these strains genetically close at the nucleotide level? And on the other hand, PanFlan would ask, do these strains have different genes or functions? Right. So PanFlan is basically pan-genome gene presence and absence uh, detection. So reconstructs the gene content profile of a strain by mapping metagenomic reads to the species full pangenome, determines which genes a strain has or lacks, produces a binary or quantitative gene family matrix, whether gene exists or not. Now, StrainPlan, how it works. So Metaflan identifies species plus retrieves marker genes. So StrainPlan only considers clade-specific marker genes. Step 2 is extract marker-based sequences. So this is basically the same mechanic. This is the same workflow that we discussed for StrainPlan. Outputs a train of strains and distance matrix and SNP counts and no functional information. However, PanFlan basically first we do first step is building or downloading the species pangenome, where a pangenome is core genes, accessory genes, and unique or rare genes. Then step two is map the reads to the pangenome. For each sample, reads are mapped to every gene family of that species. Coverage per gene is computed. Then determine gene presence or absence. So gene is considered present if the coverage is greater than a set threshold, if the breadth is sufficient, and or we have a consistent depth. Then we create gene presence absence matrix, and basically it outputs a functional genomic profile of the strain, which allows studying metabolic capabilities, antibiotic resistance genes, etc. Right. Moving on, output difference. So StrainPlan output is alignment of marker genes. PanFlan output is gene family presence or absence matrix. Right. So StrainPlan outputs evolutionary relationships, while PanFlan outputs functional differences between strains. And we discussed the StrainPlan weaknesses and strengths. Now for PanFlan, it provides gene level functional information, great for virulence or resistance comparisons, works across large strain collections, uses entire pan genome of high resolution. However, it basically requires high sequencing depth, that is a limitation. It is computationally heavy, more sensitive to noise, and requires curated pangenome, can be limiting. So that's it about this video.